Hello, everyone, and welcome to today's live broadcast, Structure and Function of Epigenetic Regulators in Human Disease, presented by Dr. Cigall Kadoch, an Assistant Professor of Pediatric Oncology at the Dana-Farber Cancer Institute and Harvard Medical School, and an Institute member of the Broad Institute of MIT and Harvard. And joining her, Dr. Chris Fry, Director of Product Development at Cell Signal Technology. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's events. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Cell Signaling Technology. Cell Signaling Technology is a private, family-owned company founded by scientists and dedicated to provide the world's highest quality, innovative research and diagnostic products to accelerate biological understanding and enable personalized medicine. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located at the lower left of your presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of this presentation. Also, please notice that you're viewing this presentation in the slide window. To enlarge that window, just click on the screen icon located on the lower right. During the presentation, you'll have the opportunity to answer some poll questions for our presenters. We encourage you to appreciate your participation, and we encourage you to do so. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use that Q&A button to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button on the left corner and follow the process for obtaining your credits. Without further ado, I'd like to introduce our two presenters. First, Dr. Cigall Kadoch. Dr. Kadoch is an Assistant Professor of Pediatric Oncology at the Dana-Farber Cancer Institute and Harvard Medical School, and an Institute member of the Broad Institute of MIT and Harvard. Dr. Kadoch studies chromatin regulation with strong focus on the structure and function of the mammalian switch-SNF or BAF family of chromatin remodeling complexes in human cancer. Her work has been centered in mechanistically interrogating rare, molecularly well-defined cancers to understand the role these complexes play in promoting a wide range of more common cancer types. Joining her, our second presenter is Dr. Chris Fry. Dr. Fry earned his PhD in oncology from the University of Wisconsin-Madison, where he examined the mechanisms of E2F-mediated gene regulation during G1 and S phases of the mammalian cell cycle. Dr. Fry then carried out his postdoctoral studies at the University of Massachusetts Medical Center, where he studied mechanisms governing the gene regulation, silencing, and chromatin dynamics during the cell cycle in the yeast, Saccharomyces cerevisiae. Chris is currently Director of the Product Development at Cell Signaling Technology, where he leads two teams, one that focuses on the development of antibodies, again, protein and non-protein targets involving epigenetics, and the other focusing on the development of products for chromatin IP. For complete biographies on these two speakers, please visit our website or click on their picture in the left-hand corner of your presentation window. I will now turn the presentation over to our speakers, starting with Dr. Kadoch. Welcome.
Thank you, Susie, for the nice introduction, and good afternoon from Boston, everyone. And a special thanks to Cell Signaling Technologies and LabRoots for sponsoring this event and for the opportunity to participate. So as some of you may know, our laboratory is interested in understanding the structure and function of large ATP-dependent chromatin remodeling complexes, most notably the mammalian switch-SNF or BAF family of chromatin remodeling complexes. And our group places particular emphasis on understanding how perturbation to the individual subunits or components of these complexes can play driving or contributing roles in human disease. So this is the large beast of a complex we study, shown here, drawn artistically to scale with its local nucleosomal environment. These complexes utilize the energy of ATP hydrolysis to essentially mobilize nucleosomes, thereby allowing for timely and appropriate gene expression. Now, before we get started, several key rules become particularly important in terms of how we think about these complexes. So the individual components are highly stable with one another in that they can resist dissociation in over 2.5 molar urea. So they're highly stable interlocking interactions. The subunits are entirely dedicated to the complex, meaning they have no monomeric forms, they don't participate in other complexes. So they're entirely dedicated to this family of chromatin remodelers. Using antibodies to one subunit or another will deplete the entire complex, and actually we've been able to use those methods to be able to uncover aspects of how these complexes are assembled in more recent studies I won't talk about today. And finally, there's no exchange of subunits in vitro. So if one purifies a complex and tries to bombard it with very high concentrations in a test tube, the additional subunit will not exchange in and out of these complexes. Now, particularly as we start to think about this family of chromatin remodeling complexes, historically they were quite challenging to study in that tap tagging or any kind of overexpression can severely alter the binding interactions and stoichiometric relationships within the complex. And so until we had the use of nice high affinity antibodies, including those by cell signaling technology, to the surfaces of these complexes, they were quite challenging to purify and study endogenously. There are 29 genes in total that encode the various BAF complex subunits. As you can see from the schematic, there aren't 29 subunits shown there, and that is because they are assembled in a combinatorial fashion, with several families having one or more paralog, but never both able to assemble into that position. A few examples of those are the two ATPases, BERG1 and BERM, also known as SMARCA4 and SMARCA2. Those are the two mutually exclusive ATPases. And another family with paralogous subunits fitting a position in the complex are ARID1A and ARID1B, which actually are the most frequently mutated subunits in human cancer. These complexes have evolved from yeast, and as you can see from the schematic, over the course of evolution, subunits have been gained, lost, and shuffled. This is likely due to the advent of things like multicellularity, linking histones, polycomb-mediated repression, and in general, a larger genome size. So these reflect the current mammalian complex view on the right-hand side of the slide with all of the subunits identified to date. Now, we're particularly interested in this complex owing to a surge of recent studies which have repeatedly revealed frequent mutations to the genes encoding the various subunits within the mammalian switch-NIF complex. And as shown in this very simplified schematic, we can estimate if we add all these exon mutations up that these mutations are frequent in over 20% of human cancers. This was actually derived from a 2013 article that we published, and this number continues to rise. Intriguingly, the complex subunit mutations appear as a broad pattern across a range of tumor types, much like that of P53. And from observing all of these mutations across these tumor types, several key themes emerge. As I mentioned, several subunits are mutated in very specific malignancies and nowhere else, so there are very clear examples of that, and we'll speak about that in this presentation. Other subunits are mutated across a much wider range of cancers, suggesting some defining commonalities between them. Intriguingly, the most frequently mutated subunits are actually not those that are required for in vitro chromatin remodeling. And this is a major problem for the field because the textbook definition employs purified complexes put together recombinantly and placed on nucleosomal templates, artificial chromatinized templates. However, as we learn from the in vivo mutational patterns here, this is likely an insufficient set of assays to assess function. And so our lab has had a major interest in understanding new assays which might be able to robustly and accurately read on the function of these complexes. The mutations also can be heterozygous, and of course this implicates gene dosage effects. So this was where we were, as the first set of 50 or so exon sequencing studies emerged onto the landscape in the field. And at the same time, a set of several studies emerged, this is in 2012, in a collection of papers discussing intellectual disability syndromes, particularly Coffin-Siris syndrome, Nicolaides-Baraitser syndrome. And as you can note in all three of these papers, all three published in Nature Genetics, the word cause was used in the title. And all of these papers implicated the genes encoding subunits within the mammalian switch-NIF complex as key players in these rare but well-characterized intellectual disability syndromes. So this was really the first time we could detect that mutations that are found here can be heterozygous, and this supports the idea that heterozygous mutations can have driver effects and haploinsufficiency gene dosage alterations within the complex. A more modern view of this, if we look at more modern TCGA data, this number continues to rise, now over 23, almost 24%. And as you can see in comparing mammalian switch-NIFR BAF complexes to the other chromatin remodeling complexes, really stands head and shoulders, and there's clearly an outsized contribution. Moreover, on the right-hand side of the slide, if we look at the somatic mutation frequencies in cases which have a BAF complex mutation, you can see that the overall mutation frequency appears to be lower than those cancers that bear mutations in these other complexes, suggesting again that these could be playing a driver or at least strongly contributing roles to those cancers. But of course, there's more than just the exons within the genome, and so if we extend some of these analyses to examine both copy number variations and adding in biallelic deletions or loss of heterozygosity, this number continues to rise, and now if we add that estimation, it's about 30% of all human cancers. Of course, if we continue to add additional categories of ways by which this complex can be perturbed, including promoter or enhancer mutations, methylation, other means of epigenetic silencing of key promoters, this list can go on, and we estimate that this is still a vast underestimation of the total percent of human cancers which have some perturbation to this BAF complex. So all of these exciting genomic results have really catalyzed a wave of interest from a variety of angles, both within our lab and in the field as a whole. Understanding complex-specific complex composition and architecture, which is one of the topics I'll talk about today, how loss of one paralog subunit can actually create an actionable synthetic lethality with its remaining paralog, trying to understand the functions of specific domains within the complex, how other factors can tether to the surfaces of these regulators to drive their targeting on the genome, etc. And this is another topic we'll discuss today in the webinar. So how complex are these complexes, really? If we now look at all the positions within the complex that have a mutually exclusive paralogous subunit able to incorporate, we get about 8 positions within the complex at which a position exists such as this. And by n-choose-r combinatorics, we arrive at over 1,000 possibilities as to how these complexes can be put together. That's quite a lot of possibilities in terms of thinking about how one might perform immunoprecipitation or ChIP-seq assays, etc. Fortunately, tissue specificity can help us out a little bit with that. So if we now take a much more restricted cell type, like a fibroblast, by gene expression, we only have about 5 positions within the complex at which one of these mutually exclusive subunits can incorporate. And so now, again, playing the same game with n-choose-r combinatorics, we arrive at 2 orders of magnitude or so lower number of possible complexes that can be formed within the cell. So suffice it to say that the combinatorial assembly, the quite complex combinatorial assembly of these complexes, gives rise to a great deal of biologic specificity. And proteomic studies by our group and others, both in a wide range of wild-type cell types as well as now cancer and other disease-specific cell types, can continue to uncover these very specific forms of BAF complex compositions. So why does this combinatorial assembly matter, and how does that dictate the biologic specificity? Of course, within the complex, there exist DNA binding domains on certain subunits, histone recognition motifs such as PhD fingers, etc. And also there are tissue-specific transcription factor binding sites across the surfaces of these complexes that have not yet been defined. But all 3 of these can drastically contribute to the targeting of these complexes. And as a whole, actually much like the field for many other chromatin regulators as well, it remains to be understood exactly how these complexes get to where they need to go on the genome. So, where we see the field, we have an unprecedented understanding of the genetic landscape of human cancer. There are hundreds of thousands now of whole exome cases. This slide can't stay up to date enough that have not been published. BAF complexes are mutated in over 20% of all these human cancers, making it the most frequently mutated chromatin regulator. And on the other side of things, we have results from high-throughput assays, omics, everything, shRNA and CRISPR screens for synthetic lethal relationships, large-scale small molecule screens, gene expression studies at a whole genome level, and genome-wide histone mark that have been deposited globally, including ENCODE, roadmap, etc. But what's really missing is the bridge between these two. And so our lab focuses on the biochemistry of these complexes, understanding their composition and stoichiometry, their structure and their topology, and understanding where they get to where they need to go on the genome by their histone mark and sequence-specific targeting mechanisms. We believe that this missing link really is a significant barrier to the identification of targeted therapeutics, and so our lab aims to focus in that nexus right there. So to try to place this missing link, we recognize that there may be opportunities in unique but well-defined rare cancer types. And so we ask the question, can we harness the power of oncogenic drivers, irrefutable oncogenic drivers, to understand fundamental questions in BAF complex biology? And similarly, could we take what we had learned about the composition of these complexes across a wide range of cell types to inform the mechanism, in this case, in a cancer called synovial sarcoma? So our goal would be if we can take these rare cancers that are well-defined genetically, translate them into a mechanistic understanding as to how they drive forward an oncogenic gene expression program, then build screens for small molecules targeting these regulators. The goal would be that we could then do this for the wide range of human patients that have a large number of BAF complex mutations. So I'll talk about a few of these rare cancers and our progress in that realm today. We originally got into this having identified this protein called SS18 as a stable and dedicated member of BAF complexes. And we cared to study this in particular in that 100% of patients that have a rare but highly aggressive form of sarcoma called synovial sarcoma bear this very specific chromosomal translocation whereby SS18 on chromosome 18 fuses in frame with SSX genes on chromosome X. And by virtue of this chromosomal translocation, a fusion protein is created, and this is fusing exactly 78 amino acids of the SSX tail onto the SS18 subunit in every single case. And so this provides strong support, of course, for an initiating role, and so our lab was to try to study these. I won't go through much of this work because it is published, but intriguingly, what we were able to identify is that the product of this translocation, the SS18-SSX fusion protein, incorporates and usurps into BAF complexes, thereby displacing the product of the wild-type allele, wild-type SS18, looking at the right-hand side of the slide, and displacing and degrading the BAF47 or SMARCB1 subunit of the complex. This led to a retargeting of BAF complexes on the genome, and for example, retargeting to a gene called SOX2 to activate that gene, and this gene was critical for the proliferative maintenance of these cells. Excitingly, we discovered that this process was reversible. If we simply change the stoichiometric balance between wild-type SS18 and SS18-SSX, we could actually shuffle between the oncogenic form of these complexes and their wild-type counterparts. So if we rescued these complexes now to take them back to those form on the left-hand side of the slide, we saw that these retargeted BAF complexes resulted in resilencing of the SOX2 gene by H3K27 trimethylation placed by polycomb complexes and a senescent phenotype of synovial sarcoma cells in culture. So this was a particularly exciting cancer example whereby BAF complexes are essentially hijacked by this oncogenic fusion. And it's brought about several new questions in the sense that it is really the first gain-of-function mechanism identified across the literature for BAF complexes. As I showed you on the earlier slide, the large majority of these are loss-of-function, gene deletion type mutations, but this is a gain-of-function. So this suggests that fusion of new proteins or domains onto these complexes can profoundly alter the targeting. Their targeting on the genome. And this targeting will recruit all the activating features of these complexes to new sites. So we were really curious if this mechanism of action could extend to any other cancers, and if so, what types and what new opportunities would this bring? So in work that was just published 2 weeks ago in Cell, we identified the EWS protein, which is a FET member, and the member of Fuse, TAF15, and EWS. These are RNA binding proteins, can actually tether to the surfaces of these complexes. And in Ewing sarcoma, the characteristic chromosomal translocation there produces the EWS-FLY1 fusion protein. We identified, as shown on the upper left-hand panel, in A673 and SKNMC Ewing sarcoma cell lines that bear the EWS-FLY1 fusion protein, that we can find that this fusion protein binds to BAF complexes as assessed by the lower panel, this IP2 BERG1 ATPase member of the complex. You can see here that EWS and FLY1, the fusion protein, are now tethered to the complexes in this setting. So we asked what happens then at a genome-wide level on chromatin. To do this, we assessed this using ChIP-seq methods, and we found that the large majority of EWS-FLY1 target sites overlapped with BAF complex sites on the genome in Ewing sarcoma cells. So you can see that this large motif that is enriched, the GGAA repetitive repeat enhancer motif, was largely shared with these BAF55 sites, as shown in the Venn diagram. An example of that is shown at the CCND1 locus, where you can see nice colocalization of EWS-FLY1, BAFM55, and HK27 acetyl. And on the right-hand side, you can see the density diagrams reflecting a very similar pattern of enrichment over the GGAA repeat enhancers. So what happens then if we perturb the levels of this oncogenic fusion in these cancer cell lines by knocking down FLI1? You can see on the left-hand side, successful knockdown of the FLI1 as shown by immunoblot. And if we now look what happens genome-wide, we see that upon suppression of the EWS-FLI1 fusion, BAF complexes are significantly diminished over these GGAA repeat enhancers, suggesting that the FLI1 component, EWS-FLI1 fusion protein, is able to recruit BAF complexes to these sites. Now, reciprocally, what happens if we put in and introduce EWS-FLI1 into naive mesenchymal stem cells on the right-hand side of the slide? We can see that if we did this, we get a major enrichment of BAFM55 sites over these GGAA repeat enhancers, as shown in the summary graph there. And importantly, we demonstrated that this is dependent on the function of the BAF complex, in that if we overexpress EWS-FLI1 and look at the RNA-seq, so these are the genes that are now activated in mesenchymal stem cells, EWS-FLI1 as a fusion is able to activate all of those target genes. However, if we pair EWS-FLI1 overexpression with knockdown of the BERG1 ATPase of the BAF complex, you can see a dramatic attenuation in this ability. Importantly, we ended up attributing this oncogenic effect of the EWS-FLI1 fusion to the prion-like activity hallmark to the N-terminus of the EWSR1 protein. So if we now look at this EWSR1 protein, they have two major domains containing prion-like features, particularly tyrosines. So if one now mutates those key tyrosines that contribute to the prion-like activity of the EWSR1 protein, we can see a dramatic attenuation in the ability to undergo phase transition as assessed in the Western blot shown on the lower left-hand panel using biotinylated isoxazole, which provides a nice readout for phase transition. And importantly, if we now look in cells by introducing either full-length EWS-FLI1 or now the mutant bearing all the mutant tyrosine residues, we can see a dramatic change in the ability of this FLI1 to recruit BAF complexes and to enable activation as assessed by HK27 acetyl over those sites. So the model for this cancer is that in the wild-type FLI1 is unable to activate these enhancers. The EWS-FLI1 fusion hallmark to these tumors, requiring the prion-like domain, activates these GGA repeat enhancers. If one makes mutations to the prion-like domain, the enhancer is unable to be activated. And finally, if we just fuse the prion-like features to the FLI1 ETS factor, you can see a dramatic activation of these enhancers. So finally, as we highlighted in the earlier part of the slides, the large majority of these mutations that have been uncovered in human cancer are a loss of function in the sense of genetic loss of function. The gene is lost. For example, in malignant rhabdoid tumor, MRT, the BAF47 subunit or SMARCB1 subunit is biallelically inactivated. We were eager to explore this, and so taking a set of cell lines that are deficient in the BAF47 subunit, we rescued BAF47 to examine the genome-wide effects. And what we can appreciate from this slide and looking at BAF complex immunoprecipitation by ChIP, we can see a dramatic increase in the global widespread targeting of this complex in every single cell line. So upon rescue of BAF47, we see a major increase in the chromatin occupancy widespread throughout the genome. And this is reflected further here with these density diagrams showing that the BAF47 subunit plays a very critical role in enabling the BAF complex to bind chromatin. And so this may begin to shed light on the larger number of human cancers that have BAF complex perturbations, as well as the various others that have different mechanisms and how we might be able to consider those. So in summary, what I've told you is that these BAF complexes are the most frequently mutated chromatin regulators in human cancer. The perturbations can result in either loss or gain of function phenotypes. Synovial sarcoma with the S18 SSX fusion was the first gain of function BAF-driven cancer that we identified, but now we know these mechanisms can extend to Ewing sarcoma and likely others. And finally, in BAF47 loss of function cancers, such as malignant rhabdoid tumor, rescue of BAF47 can restore widespread chromatin occupancy. And so with that, I'll thank the members of my lab, our collaborators on this work, and my sources of support, and you for your attention. Thank you. And now I'll turn it over to Chris Fry from Cell Signaling Technologies for the next talk.
Thank you, Cigall, for a great talk. Thank you to LabRoots for supporting our webinar. And good morning, good afternoon, and good evening to our viewers out there. I just need to flip over to my slides. My name is Christopher Fry. I'm a Director of Epigenetics Product Development at Cell Signaling Technology, and today I want to talk about the ChIP assay and really talk about things to consider when you're planning a successful ChIP assay. So I'll spend just a couple minutes briefly giving an introduction to ChIP, both ChIP and ChIP-seq, and then I want to talk about really 3 keys to a successful ChIP experiment. The first one being an optimized protocol, high-quality reagents. Next, we'll talk about cross-linking and chromatin fragmentation. And then finally, we'll talk about ChIP-validated antibodies. So what is ChIP? ChIP is a powerful technique that's used to identify and analyze protein-DNA interactions within the genome in vivo. You can use it to look at binding of histones and histone modifications, transcription factors and cofactor proteins, replication factors and DNA repair proteins. And you can use this method to look at single gene analysis using ChIP-qPCR. Here is where you would look at a handful of interesting target genes. Or you could use it and look at whole genome mapping of the protein or histone modification. Now in the ChIP assay, cells or tissues are first fixed with formaldehyde, and this cross-links histones and non-histone proteins to the DNA. The cells or tissue are then lysed and nuclei are prepared. Chromatin is fragmented either by sonication or enzymatic digestion into 150 to 900 base pair DNA protein fragments. Once that is done, the nuclei are lysed and fragmented and chromatin is isolated. We then can add antibodies that are specific to histones or non-histone proteins, and then we add protein G or protein A beads to help IP and enrich these antibody chromatin fragments from the chromatin sample. We can reverse the cross-links and purify the DNA, and then we can analyze this DNA to see what DNA we've immunoenriched using the antibody. And this can be analyzed by one or two methods, ChIP-qPCR or ChIP-seq. With ChIP-qPCR, you can design primers against a handful or so of target genes of interest and use qPCR to identify and quantify the enrichment of these target genes. With ChIP-seq, you can actually generate a DNA library and then identify and quantify and map these binding reagents back to the whole genome. Now, a poll question should be popping up on your screen. I wanted to get an idea how many of our viewers use ChIP. How many of our viewers are doing ChIP-qPCR or ChIP-seq or both? So please, if you will, select which one you're doing and let us know what type of assay you're performing. While you're doing that, I'm going to continue on with my slides. So next I wanted to spend most of my talk really focusing on 3 areas that to think about when planning a successful ChIP experiment. The first one is use of reliable, optimized protocol. Next we'll talk about cross-linking and fragmenting chromatin. And finally we'll talk about the antibodies that you use in your experiment. So when starting a ChIP protocol, you really want to use a reliable, optimized protocol, whether it be a homebrew protocol that you find that is well published or is being used by a ChIP lab, or it's a kit that you buy from a company. And at CST, we provide multiple chromatin IP kits. Here's 2 on the slide here: the Simple ChIP Plus Sonication Chromatin IP kit and the Simple ChIP Plus Enzymatic Chromatin IP kit. Both kits use magnetic protein G beads. They work with cells and whole tissue samples. They're compatible with ChIP-qPCR and ChIP-seq, and they work for histones, transcription factors, and cofactors. So really these kits both work for really, everything. You don't need a specific kit for specific protein types. These kits contain all the buffers and reagents required to do the ChIP. They have a complete detailed protocol. They're actually simpler than most competitor kits. They have DNA purification columns, which are required for downstream qPCR and NG-seq analysis. And they also contain a universal positive control histone H3 antibody and negative control normal IgG antibody. And using these antibodies in combination with the positive control primer sets really helps ensure your experimental success, regardless of whether your antibody of interest is working or not. These antibodies will tell you that your chromatin is working for an IP experiment. So next I want to spend some time talking about cross-linking and fragmenting the chromatin. So there are two methods that can be used to fragment chromatin. Enzymatic digestion, shown here on the left in blue, and sonication, shown here on the right in orange. A poll question will pop up on your screen asking which method you use to fragment your chromatin, and please indicate by checking on the box, either sonication or enzymatic. Or both, yes. So with enzymatic, cells are lysed in a low detergent buffer. We then fragment by digesting with micrococcal nuclease. And micrococcal nuclease is an enzyme that digests double-stranded DNA in between the nucleosomes. Once the digestion is complete, we briefly sonicate to lyse the nuclei, and the chromatin is purified by centrifuging out the insoluble material. Now, MNase digestion is very mild. It's done in a low detergent buffer at 37 degrees, and we actually recommend doing a partial digest. And what you get is shown here in the gel electrophoresis on the right. The second lane shows DNA that is purified from a MNase-based chromatin digestion. As you see here, you see a ladder of DNA fragments from 150 to 700 base pairs. So in orange on the right is shown the sonication protocol. Here we do a cell lysis in a low detergent buffer, and then we sonicate the chromatin, and we use acoustic energy to sonicate the chromatin into small fragments. Now, the sonication can be done in a number of different conditions. Some protocols use a low detergent buffer. Other protocols use a high detergent buffer. And different types of sonicators can be used. Traditionally, a probe sonicator has been used for ChIP, but newer water bath and ultrafocus sonicators can be used. Regardless of the sonicator used, you need to do a bit of optimization for the sonication protocol. And this is important because there's different classes of ChIP targets there are different classes of protein targets that behave differently in the ChIP assay. So there's histones, which are very stably bound to DNA, and there's a large number of histones per DNA fragment. These proteins are thus relatively easy to ChIP, and they're insensitive to harsh conditions such as high detergent and heat that's created in the sonication protocol. DNA-binding transcription factors are less stably bound to the DNA. There's fewer factors bound to the DNA fragment, and these are more difficult to ChIP. They're much more sensitive to harsh conditions. And finally, transcription cofactors, like the switch-SNF complex proteins, often do not directly contact DNA. They're often part of these large protein complexes. The antibody epitopes might be partially buried, and these are actually very difficult to ChIP and very sensitive to harsh denaturing conditions. So I wanted to ask another poll question here is what classes of protein targets are you studying with your ChIP assay? Histones and histone modifications, transcription factors, or transcription cofactors? And you can actually check more than one box if you're working on different target types. Now again, with sonication, one wants to do a bit of optimization with the protocol. And one of the important things to consider is the number of cycles of sonication or sonication time you're using in your assay. Shown on the left here is a gel electrophoresis of DNA fragments that were generated by sonication, and on the bottom we're looking at increasing time of sonication, as shown here. As you can see, as we increase the time of sonication, we see smaller and smaller fragments of DNA. And looking at this, one might think that 8 minutes of sonication might be the best because it gives us a good, nice, tight band of chromatin fragments. However, if we look at enrichment of target genes for the TCF4 transcription factor and beta-catenin, a cofactor that binds to TCF4, we see that the highest levels of enrichment for CAMK2D, a known target gene, are found using chromatins that was sonicated for only 2 to 4 minutes. We find that 8 to 10 minutes actually decreases the enrichment that we see by qPCR. So more sonication is not always better. And so what you want to do is you want to use the minimal number of cycles that you can to get chromatin around the size of 100 to 500 or 100 to 700 base pairs in length. And that should allow, if you're using the minimal number of cycles, should give you the best enrichment. Now, another thing to consider is the amount of detergent in your sonication buffer. Even with optimized cycles, if you have too much detergent, you can lose signal for some target protein types. Now, histones are very resistant to high detergent levels. As shown in this slide, we're looking at enrichment of trimethyl K27 across a region of the genome. Here we're using our simple ChIP enzymatic chromatin IP kit, our simple ChIP sonication chromatin IP kit, which has a low detergent buffer, and a leading competitor sonication kit that contains a high detergent buffer. If we look at enrichment of trimethyl lysine 27, you see that all three of these kits work equally well, giving us nice enrichment that we would expect to see in this region of the genome. However, if we look at transcription factors and cofactors, we see a big difference. Here we're looking at TCF4 and two of its cofactors that it recruits to its target genes, beta-catenin and CBP. If we look at the ChIP-seq data, here we're looking at the ACSL5 target gene. We see nice enrichment of these three proteins using simple ChIP enzymatic kit, the simple ChIP sonication kit, but using the high detergent competitor sonication kit, we see absolutely no enrichment. And this is again because the high levels of a detergent are actually denaturing the chromatin and disrupting the antibody epitopes. And so when you're looking at transcription factors and cofactors, you really want to use either enzymatic or a low detergent sonication buffer. What about tissue ChIP? Which method works best for tissue ChIP? Well, we found that both methods, the sonication and the enzymatic, work equally well in most tissues. So shown here is ChIP-seq data using mouse liver, and here we're using our simple ChIP plus sonication chromatin IP kit on the left and the simple ChIP plus enzymatic kit on the right. In the top two panels, we're looking at enrichment of trimethyl H3 lysine 4 and the FOXA1 transcription factor. And here we see very similar enrichment patterns using both enzymatic and sonication, as we would expect. At the bottom, we're looking at enrichment of ring 1B and SUS12 here at the HOXA loci. And again, we see very nice enrichment of both polycomb responsive complex 1 and 2 containing these factors at this loci. So with enzymatic ChIP, it's really important with tissues to desegregate your tissues into single cell suspension before you do the digest. And this can be done either using a tissue desegregator like the Becton Dickinson Medi machine, or by using a Downs homogenizer. But once you get that single cell suspension, it's very easy to do the digest. With sonication, the sonication itself will actually desegregate the tissue while it's fragmenting the chromatin. However, again, with sonication, we find that we have to do some additional optimization to get efficient enrichment of transcription factors and cofactors. So what we find with tissues and sonication, that increasing the cross-linking time helps out a lot when trying to ChIP for transcription factors and cofactors. So shown on the left is a gel electrophoresis of DNA fragments that we've prepared by sonication for mouse heart, mouse brain, and mouse lung. And you can see with increasing cross-linking, we do get a trade-off and we get larger chromatin fragments. However, if we look at enrichment of HOXD10 and HOXA1 by ChIP-qPCR, we see that ring 1B and SUS12, both of which bind this region, show increased enrichment when we cross-link for 30 minutes versus cross-linking for 10 minutes. And we see absolutely no increase in enrichment of this GAPDH gene, which is it's not a target of these complexes. And so we find again, with sonication, if we cross-link just a little bit longer, we can actually increase our enrichment. All right, so to just summarize the first part of my talk, tips for cross-linking and chromatin fragmentation, I'd like to remind you that I showed you that histones are resistant to harsh fragmentation conditions like heat, detergent, and the protein denaturation that's caused. Transcription factors and cofactors are sensitive to these harsh conditions. The enzymatic-based fragmentation is mild, and it works well for histones, transcription factors, and cofactors. Sonication does too, but you have to be you have to do some optimization and keep some things in mind. And that is, you want to use mild buffers that have low detergent. You want to keep your samples cold. You want to minimize your sonication cycles to really balance the chromatin fragment size and the enrichment levels of your target of interest. And finally, for tissue, you may want to increase the cross-linking time to maximize your transcription cofactor and transcription factor ChIP enrichment. For more information about ChIP, you can actually download our guide for ChIP and ChIP-seq at www.cellsignal.com/ChIP. Okay, so I want to spend the last part of my talk talking about antibodies. Antibodies are also very important for the ChIP assay. And when you can, you want to use a ChIP-validated antibody. And so a poll question will come up. I'm interested in hearing what types of antibodies people are using in their ChIP experiments. We have rabbit polyclonal antibodies, rabbit monoclonal antibodies, and mouse monoclonal antibodies. And again, if you're using more than one, you can check on more than one box. If you haven't done ChIP yet, just check on the type of antibodies that you think would work best in the ChIP assay. And while we do that, I'll move on to my next slide. So at CST, our mission is to deliver the world's highest quality research products that accelerate biological understanding and enable personalized medicine. Now, as an antibody company, this means providing the best antibodies for all research applications. And so we do extensive antibody validation across all applications, including ChIP. And when we're doing this, we use all the tools that we have at our disposal, including positive and negative cell lines, knockout cell lines, siRNA knockdown. We'll use overexpression. We'll use treatments that induce increases in protein expression or changes in protein localization in the cell. Really anything that we can do to show that the antibody is specific, is sensitive, and also works reproducibly from one experiment to the next or one lot to the next. Our products are produced and rigorously validated in-house by CST scientists to ensure consistent performance. And so for the ChIP assay, our group validates our ChIP antibodies using relevant model systems. And I just show an example here on this slide. Here we're looking at validation of the glucocorticoid receptor rabbit monoclonal antibody. Here we're testing this antibody on A549 cells that are either untreated or treated with dexamethasone. Now we know that treatment with dexamethasone induces nuclear localization of GR and binding of GR to its target genes. In this experiment, we're looking at binding to SLC19A2 and MT2A in both untreated and treated cells. And you see very nicely, we see enrichment only in the treated cells as expected. We also see no binding to this alpha satellite repeat element, which is our negative control. So here the antibody is showing exactly what we would expect to see, and that is enrichment of GR targets in treated cells. Now when testing an antibody in your ChIP assay, I always advise to titrate the antibody. This is something we do for every antibody that we test. Shown here is a titration for our EZH2 rabbit monoclonal antibody. And here, what you're looking at is looking at enrichment of the HOXA1 and HOXA2 loci as we increase concentration of the antibody in the IP assay. And you see, as we increase the amount of antibody, we do see an increase in enrichment. However, that enrichment levels off and then actually falls with increasing amounts of antibody. And so what we find with this antibody is that 2.5 to 5 microliters of antibody per IP is ideal in generating the best signal to noise, the best enrichment levels of these target genes. So with an antibody in ChIP, more is not always better. I know some people still believe that you need to use 5 or 10 micrograms of antibody for a ChIP assay, and that's simply not true. Sometimes as little as a half a microgram actually works best. And we find that it's the ratio of the antibody to the chromatin that's important. So we always titrate the antibody. Again, when selecting your antibody for ChIP, it's always good to look at validation data across different applications, as this often tells you more about the antibody's specificity and sensitivity. So here's an antibody that we have against BRG1. Again, it's a rabbit monoclonal antibody. In ChIP-qPCR, shown in the upper right, we're looking at enrichment of actually 2 estrogen receptor target genes. Estrogen receptor recruits the BRG1 switch NIF complex to target genes when cells are treated with estrogen. So here we're looking at MCF7 cells treated with estrogen, and as expected, you see nice recruitment and enrichment of BRG1 to these target genes. Shown below is ChIP-seq track, again looking at enrichment of another target gene, ENZA, and again we see very nice binding and enrichment of BRG1 at the ER binding site in this gene. On the left is shown a Western blot. Here we're actually looking at expression of BRG1 across a variety of different cell lines. These are whole cell lysates, and as you can see, this antibody really detects only a single band in these whole cell lysates. Now with BRG1, we know from mRNA expression data that A549 cells do not express BRG1. As Cigall pointed out, BRM is a paralog of BRG1 that's actually highly homologous in sequence to BRG1, and BRM is expressed in these A549 cells. So we know since our BRG1 antibody does not detect a band in these cells that it's not cross-reacting with BRM, and so we know this antibody is specific to BRG1. With glucocorticoid receptor, we already went over the ChIP-qPCR data, or the ChIP-qPCR data. Shown below is the ChIP-seq data, and again, looking at a known target gene, we see a single peak of enrichment here over a GR binding site. Very good signal-to-noise ratio. Again, looking at specificity and performance of the antibody across other applications, if we look at IF, like here we're looking at A549 cells, again, that are untreated and treated. On the left, we're looking at untreated, and as expected, you see both nuclear and cytoplasmic localization. Once we treat the cells, we see just nuclear localization, as expected, since dexamethasone induces nuclear translocation of the protein. And finally, we have a Western blot on the left. Here we're looking at whole cell lysates from 293 cells that are either untransfected in lane 2, transfected with an overexpression construct for MIC-tagged GR in lane 3, or transfected with a construct overexpressing MIC-tagged mineralocorticoid receptor in lane 4. And again, we see this antibody is very good at detecting endogenous GR, overexpressed GR, but it doesn't cross-react with their mineralocorticoid receptor protein, again showing that it's specific for GR. So I want to spend a little bit of time talking about trimethyl H3 lysine 27. Again, here we're looking at a Western blot of whole cell lysates, and the antibody detects a single band that corresponds to histone H3. In IF, we see the expected nuclear localization, and in ChIP-qPCR, we actually look at enrichment of known target genes, MyoD1 and MYT1. These are 2 genes that are repressed in these HeLa cells, and they're repressed by polycomb and repressed by this trimethyl H3 lysine 27 mark. And we see no enrichment of GAPDH or RPL30, which are housekeeping genes that are active. The ChIP-seq data is shown below, and we see the expected kind of broad binding of, or broad localization of this mark across MYT1 as expected, as this is what this is how polycomb works on its target genes. So by all accounts and different applications, this antibody looks very specific. However, we know with the histone modification antibodies that histones themselves are highly modified on lysines, both by methylation and acetylation, and these antibodies have the potential to cross-react with other methylated lysines. And so for all of our methyl lysine, acetylysine, and methylarginine antibodies, we do a histone peptide array analysis where we titrate our antibodies on an array of peptides that contain all the known modifications of histone H3, H4, H2A, H2B, and histone H1. And what we look at is we look at peptides that have a single mark and peptides that have dual marks containing neighboring modifications, so we can look at the effect of neighboring modifications on binding of the antibody. So for this trimethyl lysine 27 antibody, we see in column L that the antibody binds very nicely to a peptide containing trimethyl H3 lysine 27. However, we see no binding in K and J, which correspond to dimethyl and monomethyl, and no binding in H, which corresponds to trimethyl H3 lysine 9, which is highly similar in sequence to the trimethylysine 27. So with this antibody, again, we see no binding across peptides containing a single histone methylation site. And then over here on the right, if we look at dual sites, we see that the antibody can bind when an upstream arginine 26 is also dimethylated. Again, showing that this antibody doesn't care whether the upstream arginine is methylated. So this antibody is highly specific to K27 trimethyl. So in my last two slides, I just want to talk about the performance of rabbit monoclonal antibodies in the ChIP and ChIP-seq assay. I know there a lot of people believe that polyclonal antibodies work better for IP-based experiments, but that's simply not true. Some of our best ChIP and ChIP-seq antibodies are rabbit monoclonal antibodies. Shown here is a comparison of our trimethylysine 27 antibody against an encode-approved competitor polyclonal antibody. And we're looking at enrichment of HOX-D and MyoD loci at the top and at the bottom. And what we find is with the CST antibody, we see actually better levels of enrichment and better signal to noise than we do with the encode-approved competitor polyclonal antibody. The next slide shows a rabbit mono that we have against RNA polymerase, largest subunit, RPB1, the N-terminal domain. This antibody is used to look at total RNA polymerase binding across the genome, as the N-terminal domain does not contain a lot of the phosphorylation modifications that the C-terminus contains. And so again, we're looking at enrichment of RNA polymerase using our RPB1 rabbit monoclonal antibody and the competitor polyclonal antibody. This is a leading polyclonal antibody that is used quite often for ChIP and ChIP-seq. And we're looking at enrichment of the ZNF740 and the beta-actin genes. And again, what we find is that rabbit monoclonal antibody is superior and actually gives higher levels of enrichment and better signal to noise than the polyclonal antibody. And as expected, you see high levels of enrichment over the promoters of these genes, and then you see lower levels across the body of these genes. So finally, I'd like to summarize the last part of my talk, talking about the use of ChIP-validated antibodies in the assay. So when using an antibody, always use a ChIP-validated antibody if possible. But look at the data across other applications to look at specificity and sensitivity. Use the antibody at recommended dilutions if it's already provided. If not available, try using IP-validated antibodies, but optimize for ChIP by titration. And again, rabbit monoclonal antibodies work very well in ChIP and ChIP-seq. At CST, we have over 280 ChIP-validated antibodies, and over 50 of those are ChIP-seq validated rabbit monoclonal antibodies. For more information, you can visit our website at www.cellsignal.com/epigenetics or /ChIP, and that'll provide a lot of information about our antibodies, our ChIP kits, and epigenetics in general. Thank you.
Thank you so much, both of you, for your informative presentation. We want to now start that live Q&A portion of this webinar. If you have any questions you'd like to ask Dr. Fry or Dr. Kadoch, please let us know now. Just click on that green Q&A button at the lower left of your presentation window, type your questions into the box that appear on the screen, and click on the send button. We'll answer as many questions as we have time for. So let's get started. We have an incredible audience today, very captivated. Our first audience member has a question directed to you, Dr. Kadoch. First, Super Talk, thanks so much. I'm interested in your observation on the SS18 SSX containing BAF complex and relocalization to different loci than WT. Did I get that right? Question mark.
Sure. Thanks for the question. Yes, you got that right. In our 2013 paper on this, we demonstrated that the SS18 SSX fusion can retarget BAF complexes to the SOX2 locus. That's the published observation to date. And so this suggests that this oncogenic fusion can retarget the complex. And in looking at Ewing sarcoma, related sarcoma, with the EWS-FLI1 fusion, we saw that to be true as well at the GGA repeat enhancer sites. Hope that helps.
Great, thank you so much. Here's another question from our audience members. Since MNase digests naked double-stranded DNA that lacks nucleosomes. Won't I lose nucleosomes free region of the genome when preparing chromatin using MNase?
Yeah, thank you, Susie. No, that's not the case. We actually, and that's why we recommend doing a partial digest. As you saw in one of the slides, we always do a partial digest to give us a range of fragments from 150 to 700 base pairs. And when we do this, we have no problem enriching for transcription factors and cofactors that bind in traditionally nucleosome-free or less dense regions like transcription start sites and enhancers. So with the MNase, we still see actually really good enrichment of those regions. So no, you don't actually lose those regions.
Great, thank you, Dr. Fry. We have some great questions coming in from our audience members. Here's one. I was always told that polyclonal antibodies work best in IP and ChIP assays because they bind multiple epitopes on any one antigen, while monoclonal antibodies are limited to binding a single epitope. Can you comment on this?
Yeah, I mean that's often one belief in the field is that the polyclonal antibodies work better because they bind multiple epitopes. But these days, most polyclonal antibodies are made against a peptide of 20 to 22 amino acids. And really, when you're limited to that small of a region for an antibody epitope, the polyclonal antibody doesn't give you really any advantage over a monoclonal antibody that binds to that same region. There may be a couple different epitopes within that region that are bound by the poly, but it has the same propensity really to be buried or within the protein or exposed. And really, at that point, it depends on the affinity of the antibody. And really, in that case, polyclonals don't have any advantage over monos.
I would just say, to add to that, to what Chris said, within protein complexes, for sure, a good monoclonal that's nicely designed to what appears to be a surface-exposed epitope is often far better than a mixture polyclonal antibody where it's a mixture of exposed and buried epitopes.
Great, thank you so much. To further discuss that, here's another question focusing on the validation of IP antibodies. Why do high concentrations give less response than optimal concentrations of antibodies?
That's a good question, and that's actually something that we don't completely understand. I mean, we see that with many different antibodies. Some antibodies, the signal will increase and plateau and kind of stay the same. Other antibodies will increase and then decrease again even more drastically than the EZH2 antibody that I showed. I mean, what we do find and what we believe, is that there's the ratio of antibody to chromatin in that IP is what's important. And so once you find that sweet spot, that ratio, whether it be 2 micrograms of antibody, 10 micrograms of chromatin, really that's what's important. And as you scale your ChIP experiment, as you scale the size of your IP, you want to scale both of those the same. But yeah, I mean, that's a good question. I really don't understand why adding more antibody would decrease your enrichment.
Thank you. I want to thank our audience for being so involved. We have some wonderful questions coming in. Here's our next question. Which method of chromatin fragmentation works best for tissue samples?
So yeah, that's a very good question. I mean, we've looked at 5 or 6 different types of tissues, and we find that both methods work. Again, there are a couple, both methods work equally well, except for with enzymatic, like I said, it's really important to get single-cell suspensions before you do the digest. Once you do that, the enzymatic works really well. There are some tissues that we found, like muscle tissue, for example, where sonication actually seems to work a little better because sonication does a better job of lysing the cells and getting the single-cell suspension. The Downs homogenizer and the meta machine don't work as well. But for most tissues, with the exception of some muscle tissues, we think that both work equally well.
Thank you. And Dr. Fry and Dr. Kadoch, we have time for one more question from our audience members. Here's the last question. Do I need to use a water bath sonicator or focused ultrasonicator to get the best results for transcription factor and cofactor ChIP assays?
That's another good question. We've actually done some experiments side by side using the traditional probe sonicator, water bath sonicator, and an ultrasonicator. And what we find is that the probe sonicator actually is more consistent across experiments and across cell lines. With the other types of sonicators, we found that for different cell lines, you had to do new optimization and actually use slightly different buffers or even sonication times to get the same results. But with the probe sonicator, we were able to get consistent results from one experiment to the next and across multiple cell types and tissues. And so we've actually optimized the simple chip sonication chromatin IP kit using a probe sonicator. Yeah, so I mean, we think that works best.
Wonderful, thank you. Dr. Kadoch, Dr. Fry, do you have any closing remarks you'd like to share with the audience before we close today?
No, just a big thanks to Cell Signaling Technology and LabRoots for the opportunity. Thanks so much.
Yeah, thank you, Cigall, very much, and thank you, LabRoots, for supporting our webinar. We really enjoyed the opportunity to talk with people.
And we want to thank you both for... We want to thank you both and remind audience members that they can reach you via email. And we also want to thank Cell Signal Technology for sponsoring today's webcast, as well as LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April 2018. You will receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks for joining us. We hope to see you again here in our LabRoots virtual conferences. Bye-bye.